Hi everyone, I'm Sheena Tumutod. Welcome to Science, Technology, and Society. Chapter 3, Lesson 5, entitled Gene Therapy. So, what are the learning outcomes? At the end of this chapter, the students should be able to first demonstrate the knowledge of gene therapy and its various forms, identify and distinguish the two types of gene therapy, and appreciate the importance of gene therapy. Medical science detected many human diseases related to defective genes. Ang medical ay nakatuklas na maraming sakit na uugnay sa mga detecton genes. These types of these types of disease are not curable by traditional methods like taking readily available medicines. Gene therapy is potential method to either treat or to cure related human illness. Ang ganitong uri ng gene ay hindi nalulunasan sa pamagitan ng tradisyonal na pamaraan tulad ng pag-inom ng mga madaling gamot. Ang gene therapy ay potential upang gamitin o pagalingin ng isang tao. In 2015, a team of researchers at Harvard Medical School and the Boston Children's Hospital stated that they were able to restore basic hearing genetically made mice using gene therapy. They used an empowered therapy vector developed at the Massachusetts Eye and Ear that was identified ANCAT, which enables the transfer of genes in the inaccessible outer hair cells and introduced in the cochlea Fleisler in 2017. Gumagamit sila ng empowered theory vector na nabuo sa mata, tenga ng Massachusetts na natutukoy kapag nagaganap ng maglipat ng mga genes sa hindi na-access ng mga panlabas ng cells ng buhok at kapag ipinakilala sa cochlea Fleisler in 2017. Human gene therapy what is human gene therapy was the first realized in 1971 when the first recombinant experiment were planned. It can be simply viewed as insertion foreign DNA into patient tissues that hope successfully eradicate the targeted disease. It was that actually inspired by the success of recombinant DNA technology which occurred over the last 20 years. Ang human gene therapy as nakarealize noong 1971 nang ang unang recombinant na eksperimento ng binalak. Ito ay maaring tingnan lamang bilang pagpasok ng dayuhang DNA sa isang pasyente na tissue na Ito ay maaring tignan lamang bilang pagpasok ng dayuhang DNA sa isang pasyente na maaaring umaasang matagumpay o maalis ang target na sakit. Ito ay talagang na-inspire sa pamagitan ng tagumpay ng recombinant DNA technology which occurred over the last 20 years. The basic process, there are several approaches to gene therapy. In Fleisler 2017. Replacement of mature gene that causes disease with a healthy of the copy gene, inactivation of mature gene that is functioning improperly. Third is introducing a new gene into the body to help to fight disease. In a gene cannot be directly inserted in a human gene or cell. A gene is inserted into another gene using a carrier or vector. At present, the most common type of vectors are viruses that have been genetically changed by a carrying normal DNA. Viruses have evolved a way of encapsulating and transporting their genes to human cells in pathogenic manner, Science Daily in 2017. This is the example of gene therapy. The first one is cell with non-functioning gene. The second one is adding DNA containing a functional version of the lost gene. Third is cell functioning normally. The two types of gene therapy. The two types of gene therapy is somatic cell gene therapy and the germline gene therapy. Somatic cell gene therapy, the impact of potential and development during childhood. Prenatal somatic cell gene therapy have many concerns regarding vulnerable populations. The failure of somatic cell gene therapy may lead to vulnerable populations with further treatment. There is there is a need to avoid any harm to the pregnant woman. And the germline gene therapy, the impact on future generation, judiciary restriction on gene editing technology, legal and functional bans. The third one is there are a lot of issues regarding preclinical embryo research as they edit genes at embryo level. The last is there is a need to implement worldwide consent for a gene editing clinical traits. This is the example of somatic gene therapy and the germline gene therapy. Stem cell gene therapy are the mothers the potentials to become any type of the cell in the body. This is the example of the stem cell gene therapy. Stem cells are the mother cells that have potential to become any type of the cell in the body. One of the main characteristic of the stem cell is their ability to self-renew or multiply while maintaining the potential to ear, bones, skin, muscles, brain, and others. Ang stem cell ay mother cell na potential na maging anumang uri ng cell sa katawan ng tao. Isa sa mga pangunahing katangian ng stem cell ay ang kanilang kakayahan ng self-renew o multiply na habang pinapatili ang potential ng ear, bones, skin, muscle, brain, and etc. This is the example of embryonic stem cell. Ang embryo ay nagmula sa 4 o 5 araw na gulang na embryo ng tao na nasa blastocyst phase of development of the embryos ay usually there are several eggs are fertilized in the test tube then implanted in the human. Phase na development ang kadalasang maaring ilang mga itlog na pinapataba sa isang test tube na pagkatapos ay itatanim sa isang babae. This is the somatic stem cell. This is the somatic stem cells for example. Blood vessel, teeth, bone marrow, liver, heart, gut, skin, brain, skeletal muscle. Somatic stem cell don't exist throughout the body after embryonic development and are found inside of different types of tissue. These stem cells have been found in tissue as the brain, bone marrow, blood vessel, skeletal, muscle, skin, and the liver. The biotics of gene therapy. The biotics gene therapy, there are ethical issues involved in gene therapy. Some of the injuries cited are first one is how can good and bad uses of gene therapy be distinguished? Who decides which traits are normal and which constitute a disability or disorder? Will the high of cost gene therapy make it available only to the wealthy? Could the widespread uses of gene therapy make society less accepting of people who are different? Another controversy moves the germline therapy. As discussed, germline therapy is genetic modification of germ cells that will pass the change on the next generation. There are a lot of questions on the effects of the gene alterations to the unborn child and to the next generation, since the alteration can be passed on. In the United States, the government does not fund the researches on the human germline gene therapy. And this is the germline gene therapy example. There are a baby with genetic disease, healthy baby, genetically improved embryo, and the cell culture, genetically modified cell from the culture. And this is the example of the other example of germline gene therapy. And that's all for today. Thank you.